Cool. So we are live. So today's session is on topic 2. So this is everything about the structure of nucleotides, DNA, the structure of DNA, and DNA replication itself. And also other uses for nucleotides such as ATP. And then there's also a bit about water towards the end. It's about 1 page. So, let's start. Is it working? I don't think so. Give it a good quick look. I think it should be working now. Cool. So, nucleotide is a molecule that contains 3 parts. You've got a pentose sugar, so that is this main central bit here. Then you've got this nitrogenous base up here, and then you've got the phosphate group over here, the blue circle. So, these form your nucleotide. The pentose sugar can either be ribose or deoxyribose. Ribose being found in RNA, where the RNA comes from, and deoxyribose found in DNA. There are 5 types of nitrogenous bases: adenine and thymine, and cytosine and guanine, and lastly uracil. Those first 4 are found in DNA. The last one, so uracil, is also found in RNA, replacing thymine. We'll get onto that later. These bases are found in pairs, so A and T, and C and G. So often you shorten them to A, T, C, G. So those pairs are held together by hydrogen bonds, and the first pair has 2 hydrogen bonds between them, third pair having 3. DNA is a structure made up of 2 chains of polymers of these nucleotides. The formation is by a condensation reaction between this phosphate group up here and the pentose sugar. And this bond here is your phosphodiester bond, and it's quite important you know that. It comes up quite a lot. It's 1-mark questions. And this bit here is your sugar phosphate background. If you look, this one here, these pentagons are pointing downwards, these ones are pointing upwards. This is because DNA is anti-parallel. It's parallel because they're both going along, and then anti because one's going in the opposite direction. This isn't too important for now, but you do have to remember that it's anti-parallel, especially when drawing it, that these point in opposite directions. And lastly, these 2 pairs, these 2 polymer chains are held together by the hydrogen bonds between the bases. So as you mentioned, the 3 and 2 bases between your GC and AT pairings. So that is showing them there with the dashed lines. So how does DNA relate, its structure relate to its function? So first thing, it's very stable. So it can withhold the genetic information between generations and not have significant change. The next one is your hydrogen bonds. They're relatively quite weak, so they can be easily pulled apart for DNA replication or the production of RNA. The base pairing also helps with the structure, allows you to replicate properly, allows that to make an exact copy, the production of RNA. So large molecule, so it is thousands, hundreds of thousands of bases long. I think it's even millions, containing 20,000 different genes. So it's a lot of genetic information, so it has to be quite a long molecule. And And the sugar phosphate background, but again quite strong, helps protect those inner bases from changes which might occur. So how is RNA different? RNA is another polynucleotide. There's a polymer of nucleotides, but uses ribose instead of deoxyribose. And it also doesn't have the base thymine, instead uses uracil. So uracil acts exactly the same as thymine, forming the same complementary base pairing with adenine, again with those two hydrogen bonds, but it's just in RNA, it's just different. So RNA is normally single-stranded, although it will sometimes fold onto itself, such as later on you will learn about tRNA. So it's a type of RNA used in protein production. We'll get onto that at topic 8. And it can also work as a catalyst, such as in ribosomes, which we'll also talk about later, probably topic 3. So not this week, next week. So semi-conservative replication. This is the theory or the model we use to describe replication of DNA. So first thing, we have our structure. It is then unzipped. This is done by the enzyme DNA helicase and forms 2 separate template strands. Free-floating nucleotides will then bind to their complementary base on the template strand. So then you have, so all of these ones here, they will then bind, and then something like DNA polymerase will catalyze the bond or the formation of the phosphodiester bond between your nucleotides, and that will then form your new strand. So, how do we know this? Where am I? Okay, so we have done an experiment. Scientists have, I think it's Watson and Crick who did it. They grew bacteria in heavy nitrogen or nitrogen 15. And so when they looked at the DNA, it's all heavy and all sunk to the bottom. Then they took some of this bacteria and then they grew it in light nitrogen or nitrogen 14. So all the new nucleotides being produced are from this light nitrogen. So we can see what happens. So when they looked at the DNA for this one, they saw a band in the middle. This means that there's going to be a mix. So it's not this dark brown colour at the bottom, and it's not this light green one at the top. It is in fact this sort of yellow mixed one in the middle. The colours are just for demonstration here. So when it replicates again, the template, this one here, is going down to here, and then a new one here is made. Or this one is going to form a template again, and it's going to mix again with the new nitrogen for the second generation, forming this green band at the top. This green band here is this one here. This yellow band here is this one here. So the way this proves the semi-conservative is it disproves other theories. So if it's conservative replication, where in which the daughter set of your daughter genome or daughter double helix contains both strands from the parent, then we'd expect 2 bands here, where in which all of this is conserved, and then a whole new light green one is in this generation. Dispersive replication is when the daughter helix is made up of a mixture of parental and new DNA. And so you might get something like this for the first one, but then in the second one you won't get these 2 distinct bands. You'd get each genome, so in this case you have 4. It hasn't shown it in this one for sort of slimness, but you get 4 distinct bands where each one is, as opposed to 2 very discrete bands. So ATP. ATP is our sort of unit for energy. So anything that requires energy goes through ATP. So it is used in your metabolic processes. So any sort of breakdown where you require energy, it's going to be using ATP. So in this case, we're making something. So turning amino acids into proteins, like we talked about last week, that requires ATP. Movement also requires ATP. So your contraction in muscle fibers, you will look at that in topic 14, I think, how the muscles contract, and they require ATP in the filaments. Then you've also got active transport. We will be talking about this soon. So your sodium potassium pump, that requires energy, and it does this by the binding and use of ATP. So ATP binds to the pump, and that gives the energy to move these ions across the membrane. For secretion, so to form these vesicles and the contents of the vesicles to then be released out in the cell, and then also the activation of molecules. So when you use ATP, you will remove a phosphate. This phosphate can bind to proteins and cause them to become active. So the structure of ATP, or adenosine triphosphate, is its full name. So it is a nucleotide, as shown here. Again, with this ribose sugar, so the same one as RNA. Always contain adenine, so don't say adenosine for your base. Very easy to confuse adenosine and adenine. So adenosine is this whole ribose adenine structure, whereas adenosine is all of it, adenine is just the base. And it also contains 3 phosphate groups, as shown there. So these, when it's being used, it is hydrolyzed, so you add water to it, and this will produce ADP, so adenosine diphosphate, so it's lost a phosphate, and then also produces this phosphate. So like we said in the previous one with activation of like enzymes, that phosphate there is the one that will go off and bind those proteins to those enzymes to do stuff. So this will also be easily reversible, just your same reaction but in the opposite way. So that'd be your production of, your production of ATP, yeah. So last slide is very short one, didn't have too much time to prepare. Next one should be longer. There's no questions at the moment, nothing from the last live stream in the week since. So we have water. Water is very important for life. As a metabolite in reactions such as hydrolysis. So when you add water to break support, but also the production of water. So that happens in condensation reactions. As a solvent, so it allows for aqueous ions and lots of other stuff to dissolve into the water so they can react. And cohesion and surface tension. So this comes up when we talk about plants, but this comes from hydrogen bonding. So it's a special type of bonding between water molecules, and it allows them to stay together and form this surface tension, which allows this mass movement of water. To think of like blood, also the xylem is mainly where it is. So, also the idea of the high latent heat capacity. The evaporation means high latent heat high latent heat of vaporization, or vaporization or evaporation means water is lost much more slowly, whereas the high specific heat capacity will buffer sudden temperature change. So it means that the water takes a lot of energy to heat up, and it loses a lot of energy cooling down, so it takes a lot longer. So here we've got some questions to go through. So let me just look at this. So, what is the name of an enzyme using DNA replication? So, we are using, what do you want to think? So, we're using DNA helicase that splits our 2 strands into the template strands, and then DNA polymerase. Use your knowledge of semi-conservative replication to suggest the role of single-stranded DNA fragments and the role of the DNA nucleotides. So, the role of the single-stranded DNA fragment, so that is your template strand, and it allows for the complementary free-frame nucleotides to bind to it, acts as a template, and then the role of your DNA nucleotides, that is to bind your template strand and then form your phosphodiester bonds between each other to form your new strand. So we've got the idea of DNA polymerase. Okay, if I had read it more, DNA polymerase is the one you want. So, that's the only thing I didn't say of it determining the order. So I should have said that, but yeah, AQA template determining the order of the bases, and then forming your base pairs. So describe how ATP is formed from component molecules. So it's talking about what it's made up of. So the idea of adenine, ribose, sugar, and then 3 phosphates. How those phosphates are made up by a condensation reaction. So the loss of water. I think that's it. And the enzyme used is ATP synthase. So quite often, if you don't know an enzyme and it's making something, synthase is a good example. 5 properties of water. So these are the things we went through. So I'm just going to look at it. So the idea of it being a metabolite in condensation hydrolysis reactions, as a solvent. So reactions can occur because if you don't have it as a solvent, stuff is locked up in either solids in its solid state or a gas state, and so then they can't really react. High specific heat capacity, again, the buffers to change temperature, large latent heat vaporization, provides cooling effect. So that is not what I said previously, but it's along the same lines. Cohesion supports column of water. So this is stuff we'll talk about later. Provides surface tension support for animals. That is something I didn't mention. So the idea of the surface tension, so things like bugs can go across it. So that is it. There's no questions. I'm going to end it here. See you next week.